Good morning, students, and today we will continue the in the chapter Human Reproduction. So in this, the last class we discussed the cleavage pattern, how the cleavage takes place and what is the cleavage, types of cleavage, and in our human beings how the cleavage process takes place. And here we will see that in the first step of the cleavage, we already discussed that the formation of morula. Formation of morula. So this morula is a solid mass of ball which at this stage it enters into the uterus. So here if we will see the morula, so as if it is a fallopian tubule of the female reproductive system, and this is the uterus where endometrium is present. And here this morula stays, which is a solid mass of the cells, and it enters into the uterus. And here as soon it enters into the uterus, so it started to undergo that some changes, and still it is structurally we will see that morula, it's already undergone compaction process. So what is that compaction process means here? As we already discussed that here the two types of cells are produced. So smaller cells and the larger cells or bigger cells. And here the smaller cells that they compact. You see, the smaller cells form an outer layer and the bigger cells are restricted in the center. So these are the bigger cells, and this rearrangement of the cells in the morula is known as compaction. And here as a result of this compaction, here the outer smaller layers smaller cells are arranged, and inner here the bigger cells are arranged, and this stage now it enters into the uterus, and once it enters into the uterus, and it is just, if you see that it is an endometrium, and here now it is the morula. In the endometrial lining, this morula is incorporated. So now when this, now we will take only this part. This is the endometrial lining, and we know that so the endometrial lining, which is highly vascular and glandular. So it is highly vascular. That means it supplies with a large number of blood vessels. So it is a vascular and glandular. The secretions of these glands which are present in the endometrium is the source of nutrients for the developing embryo. So this gland secretes some substances which helps to the nourishment of the developing embryo. So that's why this endometrium layer is highly vascular and glandular. And outer side of this endometrial lining, because it is just a membrane, and outside this is a fluid is present. This fluid is present. So now here this is the morula which is present in the endometrial lining. This is the morula, and this is the structure where exactly when it enters into the uterus. That means the morula which enters into the uterus. And here the next process, if we will see that once it enters and it incorporated in the morula. So the next process as we are taking here, it is the formation of blastula. Second step, formation of blastula, or this blastula is also known as blastocyst. Blastocyst. So here what happens exactly here means, this is endometrial fluid, it enters into the morula. So for that there should be some space. For this fluid there must be some space in the morula. So now what happens here means that outer layer, and this outer layer, the cells are flat cells. The cells are flat cells, and this layer, the smaller layers, already we know that in the morula, due to the compaction, the smaller cells become outside and the larger cells are in the center. So here these smaller cells are flat cells, and this layer, this entire layer is known as trophoblast. Trophoblast, or it is also known as trophoectoderm. Trophoectoderm. And then the bigger cells which are present in the center of the morula, they move at one end of the morula, or this structure. In the morula, entire central part is filled with these cells, these larger cells. But in this blastula or in this blastocyst, so here these all the cells are moves at one end of the trophoblast, and this is known as now embryonal knob, or embryo proper. See here, these are the bigger cells and these are the smaller cells. Smaller cells makes the one single layer with the flat cells, and which is known as trophoblast or trophoectoderm. And the bigger cells are arranged at one side, and which is known as embryonal knob or embryo proper. So here if you will see that the cavity appears in this cell. At this stage, the cavity which is present in this blastocyst is known as blastocele. So like this, the blastocele is formed by which from which the mouth or anus. So it is according to the either it is protostome or deuterostomes. Either blastocele forms the mouth first or anus first, and this blastocele is formed, and which is a cavity present in the blastocyst. And here this embryonal knob or embryo proper is actually that gives rise to the embryo. So that trophoblast, which are having small cells, it does not involve in the formation of embryo, but only these bigger cells and embryonal knob or embryo proper, it gives rise to the embryo. So from these cells only that embryo is developed. But trophoblast the formation of extra embryonic membranes like chorion. So later we will discuss about this extra embryonic membranes. So but here this trophoblast or trophectoderm does not involve in the process of embryo formation. Only this embryonal nerve involves in the process of formation of embryo. And this blastocyst, now this fluid, so here these fibroblasts are present. Fibroblasts. These are known as fibroblasts because here the maternal blood is present. So that means mother's blood is present here. So in that the fibroblasts and all present here. So and one more thing here, it is that the cells which are cells of the trophoblast which are contact with the embryonal nerve cells. See here, if you will see, here I will draw neatly. This is the trophoblast layer. Trophoblast layer. And here the cells of embryonal nerve are present. This is the embryonal nerve. And here if you will see, these cells which are contact with the, so these are the cells. Which contact with the embryonal nerve are known as cells of Robert. Cells of Robert. So the cells of Robert means the trophoblast cells which contact with the embryonal nerve or embryo proper. These cells are known as cells of Robert, and this is the structure of blastocyst. So here the structure of blastocyst contains mainly 4 parts. One is cells of Robert and trophoblast layer, then embryonal nerve and blastocyst. And the side which the embryo proper is present or is located, so this end is known as embryonal pole. It is known as embryonal pole where the embryo proper is present. So this is embryonal pole. And opposite side of the embryonal pole is known as abembryonal pole. This is the embryonal pole where the embryo proper is present, and opposite pole is known as abembryonal pole. So actually here is the embryonal proper and here is the blastocyst is present. And this blastocyst, it forms either mouth or anus. So if it forms first mouth, these type of animals are known as protostomes. Stoma means mouth, and where the mouth is formed first from this blastocyst, or it is actually this entire blastocyst is that the alimentary canal is formed from the blastocyst, and from that blastopore. Blastopore is formed later in the later structures, and from that the mouth or anus is formed. On the basis of that, either these animals are categorized by either protostomes or deuterostomes, and this is the structure of the blastocyst. And the next, if we will see that the next process here, it is implantation. Implantation. See, in this implantation, what is implantation? Means the incorporation of this blastocyst into the at the morular stage itself, it is incorporated into the endometrium lining of the uterus. So this is the endometrial lining, and this incorporation deeply into the endometrial lining of the uterus is known as implantation, and this implantation takes place, it's about 6th to 10th day. 6th to 10th day. So as we already discussed that the morula formation, so from that morula formation it takes about 3 days, and after that 3 days, once the morula enters into the uterus, then again 4 days. It takes again 4 days for the formation of blastocyst and for the incorporation deeply into the endometrial lining of the blastocyst, of the uterus. So total it is about 7 days on average. But actually it is 6th to 10th day, we will generally say that, but average day or average period for the implantation is 7th day. It is from the fertilization day. So starting from the fertilization, it is about the 7th day the implantation takes place. And how this implantation takes place? So here the endometrium now it completely occupies, so it is deeply penetrates. So once if we will see that if it is the uterus and the lining, due to the secretions of hormones like progesterone and estrogen, the endometrial lining is fully developed or completely it's developed and highly folded, which is present in the uterus. And here, see for example, here if the implantation takes place, so now this fluid which is present around the endometrial layer helps in the implantation process, and here one reaction is formed. So that reaction is known as decidual cell reaction. Decidual cell reaction. So up to this stage, actually corona radiata is already gone, so as a egg membrane, and another membrane, that is zona pellucida, still it is present in the blastocyst stage. Once the fluid from the endometrium it enters into the blastocyst, the blastocyst size rapidly increases. The size of the blastocyst increases. As this size increases, the zona pellucida is also dissolves. So here these type of reactions are known as decidual cell reaction or DCR. What type of changes takes place during these decidual cell reactions? The first one is changes in DCR, that means decidual cell reaction. So in the decidual cell reaction, the first one is permeability. For blood vessels, permeability for blood vessels in the endometrium, increases. So the permeability for the blood vessels in the endometrium, it increases. And the next one is these fibroblasts which are already present in this fluid, so the mother's blood, and these fibroblasts filled with glycogen. So these fibroblasts are completely filled with glycogen. So the second change during this decidual cell reaction is the fibroblasts become filled with glycogen. So now these cells are known as glycogen filled cells. So these cells filled with glycogen completely, and these cells are known as glycogen filled cells. Glycogen filled cells. So the fibroblasts filled with glycogen and they forms the glycogen filled cells. And the next one is the third one, endometrium gets disrupted. Endometrium gets disrupted. Means here at this at the area of the cells of Robert, so here this endometrium gets disrupted only at that place. So because now the next stage, so first next stage, okay, what happens then in the next stage, this trophoblast layer, it gives rise the finger-like projections deeper into the endometrium. So this gives the finger-like projections deeper and deeper into the endometrium. For that incorporation or for that projecting these finger-like projections, here at this area the endometrium gets disrupted. So now these are known as chorionic villi. These finger-like projections which are developed from the trophoblast. So it is very important. The chorionic villi are formed by the trophoblast layer. Chorionic villi. So these chorionic villi are formed from the trophoblast. And this is the next stage. And here in the human beings and apes, the type of implantation is interstitial type. Interstitial type of implantation takes place in human beings and apes. So in these two types of species, the type of implantation is interstitial. So what is interstitial type of implantation means? So already we discussed the maternal fluid around the endometrium. And now these finger-like projections which are formed or developed from the trophoblast layer, they are incorporated into this fluid. So this is known as, this type of implantation is known as the interstitial type of implantation. And this process, it's complete, for this implantation process, it takes place nearly 7 days. So after the 7th day or 6th to 10th day, the implantation takes place. So here, this chorionic villi now it forms or it makes the fetal placenta part. So in the placenta we will discuss in detail about this placenta parts. So and here this endometrial lining and maternal fluid, so that is from the blood vessels from the mother, it forms, actually placenta has two parts. One is fetal placenta and maternal placenta. Fetal placenta is formed by this chorionic villi. So chorionic villi later it develops into the fetal placenta. And maternal placenta is formed by the blood vessels and fluid which are present around the endometrium. So this is from endometrium fluid. And through this maternal placenta, the nutrients which are passed through this placenta to the fetus. And fetal placenta, it receives the nutrients from the mother placenta, and also it gives the carbon dioxide and other waste materials which are produced by the fetus. So later in the placenta topic we will discuss about these types. And here up to the formation of placenta, these chorionic villi are the source of the receiving mother's nutrients for the developing embryo. And also here the one more source is the fibroblasts, already they are filled with glycogen. So that is also one source of the nutrient for this developing embryo. And these endometrium also becoming thick and thick due to the continuous secretions of the progesterone and estrogen. And we know that progesterone is very important for the maintenance of pregnancy, because this progesterone makes the endometrium which is suitable for the implantation process as well as for the developing embryo. So for the proper incorporation and proper position of the embryo within the uterus, this endometrium should be in proper thickness. So that maintenance of the endometrium is only done by the progesterone hormone. So this is how the changes takes place in the DC, or this is the third step, that is implantation. So in this implantation, so these changes takes place. So that changes and that is reactions are known as decidual cell reaction, and the changes, one is permeability for blood vessels in endometrium increases. So because it should be supplied with a large number of blood vessels for the developing fetus. And next change is fibroblast filled with glycogen. And now these cells are known as glycogen filled cells. The next stage is endometrium gets disrupted, and then only this chorionic villi are deeply projecting into the endometrial lining, and then chorionic villi are formed from the trophoblast layer. So especially at the area where the embryonic proper is present. And then the type of implantation in human beings and apes is interstitial type of implantation. Reason, these chorionic villi are incorporated deeply into the interstitial fluid of the endometrial layer. So that's why this is known as interstitial type of implantation. And the next process in this embryo development is gastrulation. So the next stage of blastula is a gastrula where exactly the differentiation takes place. Differentiation takes place, that means up to here we don't know from which cell which organ is formed under. So that's why it is known as indeterminate cleavage. And once the blastula stage completes, the next stage, that is gastrula, where the 3 germ layers are formed. So the next process is gastrulation. And here in this gastrulation, what is the process of gastrulation? Means conversion of blastula into gastrula. So this gastrula now it forms when it is known as gastrula means when it forms the 3 germ layers like ectoderm, endoderm and mesoderm. So from this the 3 germ layers are formed, and we have to remember that one thing, these 3 germ layers are formed from the embryo proper only. So now you just forget about the trophoblast layer, only here the embryo proper gives rise to these 3 germ layers which is known as the gastrula. And this entire process, how this blastula converted into gastrula, that process is known as gastrulation. And here, so if you will see that the trophoblast layer, and here this is the blastocyst. So just I will draw the diagrams how the blastula converted into the gastrula and which gives rise the germ layers. So here trophoblast layer, and here these are the chorionic villi. These cells gives rise chorionic villi where the embryo proper is present. Embryo proper is present. And here from this embryo proper, and first what happens exactly means these cells of rubber get dissolved. The cells of rubber and remaining as usual remaining trophoblast is present, but only here the cells of rubber get dissolved. So here this is the area where the cells of rubber are present, and here they dissolve completely and remaining trophoblast layer is as usual. embryo proper is present. Embryo proper is present at here. And then, so this is the A stage and this is the B. And now in the C stage, this embryo proper moves. So just it moves upside or it moves towards the embryonal pole. So this is the embryonal pole where embryo proper is present. So now these embryo proper cells become visible, and here the trophoblast cells are present. So these remaining trophoblast cells present as usual, and here this is the embryonic proper or embryonal nerve. So and this now, this formation of the disc, so this is the discoidal structure. Discoidal structure, see where exactly the embryo proper is also moves upwards, and this process is known as delamination. Delamination. So in the delamination process, what happens? This embryo proper now it divides into two different types of layers. So what are that layers means? The upper columnar cells, the larger and the columnar cells, and here this is the trophoblast layer, as usual it is present, and lower, so this is the D stage, and here the discoidal structure has formed in the C, and now from this the lower cuboidal cells are formed. Upper layer is columnar cells and lower layer is cuboidal cells, and this upper layer is known as epiblast. Epiblast, and the lower layer is known as hypoblast. Epiblast and hypoblast. And now in the next stage, actually what happens in the E stage? In the E stage, the first germ layer, that is endodermis or endoderm. Endoderm is formed from these epiblast cells. So these epiblast cells, how these epiblast cells are formed means, from this epiblast, the cells which detached from this layer. So here in this stage, the cells which are detached from this epiblast, and this process, detachment of these epiblast cells, are known as delamination. Delamination. And here, due to the delamination process, and now if you will take this layer, this is the epiblast layer, and here is the trophoblast. And now these delaminated cells, they form, so here complete trophoblast layer I will draw. So now from this, it undergo the mitotic divisions. There is cells which are delaminated from the epiblast, they continuously undergo the cell divisions, that is mitotic cell division. As a result of this, the cells are formed in this manner. and now this is known as the endoderm layer. The first germ layer which is to be formed is endoderm. It is very important to remember that the first germ layer is not epiderm, but the first germ layer which is formed from this embryo proper is endoderm. So like the first endoderm, next in the next class we will see the remaining two germ layers, how they form, and that means we will continue with the gastrulation process.